to investigate the role of GULP/CED6 gene in EADME signaling in cellular efferocytosis and immunosurveillance. Cancer research, much like cancer, is constantly evolving. Scientists are working tirelessly to uncover the mechanisms that turn normal cells into cancer cells. Additionally, great strides have been made in the study of genomics and proteomics that has given the scientific community a lot of confidence. The goal of this study was to explore the GULP/CED6 genes and its role in presenting cancer cells to scavenger cells. H. Banerjee et al., 2016. Modern cancer researchers are very interested in the genes associated with cancer and the effects of their products. Modern uses of CRISPR-Cas9 technology may one day be able to target these genes therapeutically. In this study, CED6 siRNA was used to knock out the GULP/CED6 gene in astrocytoma, HTB12, cell lines to study the effects on the expression of EADME signals. H. Banerjee et al., 2016. Cancer cells proliferate out of control if they are not able to transcribe EADME signals. As the name suggests, these signals mark cancer cells for disposal by scavenger cells. Cellular material from cancer cells may cause damage to surrounding cells and inflammation, so scavenger cells are like a biohazard team hired for a deep clean. This study found that astrocytoma, HTB12, cells with CED6 knockout did in fact have decreased EADME signal activity, therefore, CED6 was discovered to play a role in cancer cell clearance. H. Banerjee et al., 2016. The implication of such studies places cancer researchers a step closer to the development of novel drugs for the treatment of the proliferation of cancer cells. Cells die naturally to keep tissues healthy in highly regulated processes like apoptosis and ferroptosis. If cells fail to die, then cancer proliferates. Luckily, cancer cells do not have to die to be targeted by scavenger cells because of surface expression of EADME signals. H. Banerjee et al., 2016. Future drugs may be able to induce the expression of proteins that target cancer cells for destruction. Studies such as this highlight how cancer cells hide from the cells that wish to destroy them. These fugitive cells proliferate, causing mild effects like inflammation, or more devastating effects like death. In the future, cancer cells may be induced to express EADME signals. H. Banerjee et al., 2016. Also within this study, GULP genes were discovered to play a role in modulating TGF-beta signals in ovarian cells. TGF-beta induces cell growth arrest, however. The cells are not sensitive without GULP present. H. Banerjee et al., 2016. More research will be done in the discovery of novel genes associated with cancer proliferation. These mechanisms will help scientists design drugs that induce the upregulation of these genes in cancer cells as well as in macrophages. H. Banerjee et al., 2016. The elimination of cancer cells will be a new frontier in human history. References: H. Banerjee, B. Bazemore, A. Barfield, D. Crummity, C. Krauss, G. Payne, J. Rousch, and V. Manglik, 2016, 1130. A study to investigate the role of GULP/CED6 gene in EADME signaling in cellular efferocytosis and immunosurveillance. Retrieved from ncbi.nlm.nih.gov. https://www.ncbi.nlm.nih.gov/pmc/articles/pmc 5345489